Let's continue from the session 5 and 2, which is the experimental design. What we are going to learn in this session, we are discussing the experimental design and method for association test, and also talking about the reproducibility and genome-wide and multiple hypotheses for the genetic studies. And also we are finalizing the entire session with the discussion of challenges in association testing. Okay, so the first topic about the experimental design of genetic association study. We have several things to consider in a experimental design of genetics association study. So first one is the research question, obviously, and most of the genetic studies in these days, we are asking about the genetic architecture of disease or trait. And what you have to do is to understand the samples or cohort we are using. And this is obviously related to sample size and statistical power. And also you have to think about the method or the methodology to test hypotheses. And each methodology, which is the statistical evaluation, they have pros and cons, and you have to understand what will be the best solution for the certain experiment or the certain samples. And also you have to think about confounders. And lastly, what you have to understand is the reproducibility. So whether you can replicate findings, and also the experimental method also should be replicated. And also think about the standard notation, because we have to discuss the result from multiple different studies, and has to be coming up with the unified format, and then you can easily compare between them. And all of these things considered in certain experimental design, then we call it robust. So robust means that it's a higher reproducibility and also high credit for the research, then everyone can agree with this experiment. So the most of the case for experiment design for genetic disorder following this notation. So from the study population to genotyping and association test and post-hoc analysis. So study population is obviously, in our case, is we are using the biomedical samples. So mostly the certain participant in your samples probably with the diagnosis, right? So you set the hypothesis for certain disorder, then you set up the research question. So, even though you set up the same disorder as previous study, but you can obviously set up different questions for your research, right? So sometimes you can ask certain, types of the variants or certain types of the genes, or certain clinical variables, right? So you have to explore the things you can ask in your experiment, right? And then you can consider a component in a genetic architecture. So we know the many components in a genetic architecture, like the types of the variants, or types of the the allele frequency of the variants. So all of these things can be considered in your experimental design. Then you can pursue whether those things are severely affected on your experiment or not. Okay, and also what important things in most of the study is you have to explore a previous studies, right? So you can look up some, the previous studies, and then think about how to, connect your ideas to the previous evidence, right? And things like, the rare variants associated with certain disorders, or common variants associated with certain disorders. And in this evaluation, what we can do is look at their effect size, like, so you can basically look up their penetrance or look up their the level of association and how many people having certain types of mutation in the samples of your interest. Okay, and regarding samples, what you can think about is whether you are comparing your samples with control sets. So that is the typical experimental design we call case control samples, or you can compare your genetic factors within families, right? So in that case, we say family are designed for the experiment. And also, when you talk about your samples, you have to make sure how to select your samples in your experiment. So you have to suggest the criteria for sample collection. And this criteria, we can also has to containing the phenotyping method, right? So for instance, you are doing the depression disorder research, then you have to you have to make clear about how to define depression, right? So you can either use the clinical samples, or you can use the self questionnaire, so like people can do the survey about some questions, how the many days you feel depression in a month, or how much you feel depression in a certain situation. So in every genetic studies, they have their own phenotyping method, and you have to look up their strategy and also implement your exercise as well. And also lastly, you have to think about the sample size, right? So sample size ensure you the statistical power, and that makes you the better experiment design in terms of the confidence of your research. And also lastly, since we are working on a genetic studies, and even, especially in these days, the genotyping platform is the critical things you have to make a decision. So you can go, either on exome sequencing for your design, but also you can use the whole genome sequencing. And these things all, has some balance between your the budget and your capacity to conduct the research. So all of these things also you have to do some benchmarking of what is the good things in your experiment or not. Okay, so since you set up the experimental design and the population, then you can move on to the genotyping, right? So actual genotyping exercise. So you basically generate the genotyping data set and also think about their pipeline, and also the things you must consider is the quality control. So the data set is always has some the up and down in a measurement process. So you have to make sure all the good things coming in your data set, and that process we call the quality control. So make sure you have all good genotyping data set in your samples, and also you can compare those data set, the quality information with the previous studies. Then you can reckon that whether it is the good data set or not. Okay, so so once you finish genotyping, then you move on to the association testing. So for the genetic association, you can choose some different ways. So you can test the association based on the locus. So you basically compare the locus of the variants, or you can compare the genes, right? So you can compare the number of variants per gene between case and control. Or you can compare the category, right? So category is like, one example is like a variant consequence. So you can compare the stop codon mutation category versus missense category, right? So how many nonsense mutation arising in a cases or controls, right? So you can basically compare the category of certain variant types between the cases and control. And also variant types has to be considered as well. So you can basically compare inherited variant or the novel variant, right? So, I mean, now we know about the session 2 and 3, some terminology. So you now connect the idea between the fixed size and inherited or the de novo variant, right? So in a genetic association testing, you are considering the factors like a transmission, and also you can also implement the allele frequency in an association testing. So the situation like you're comparing the common variants or the rare variants, it has to be different because the common variants is you can easily use the per locus test because the common variants is commonly observed in across the many of loci, right? But rare variant is rarely happening multiple loci because I mean it's literally rare variant. So not many people has the variant in a certain locus because of the rarity. So in that case, you can consider to use the per gene test or per category test because you can basically summing up the numbers from the samples of the rare variants, right? Then in association testing, what you have to consider is the confounders. So you can think about the genotyping quality and batch effect. So batch effect is the differences across the data set. So think about the situation like, you do the genetic study of type 2 diabetes, and you are looking at the data set from the Korea University Hospital, and the sample size is the 2,000 people, 2,000 patients, right? It is good data set, but you might want to increase the statistical power by increasing the number of patients, right? So you are visiting other hospitals like Seoul National Hospitals or Samsung hospitals, and you collect additional 4,000 patients for your study. So now you have 6,000 patients, but these 6,000 patients collected from the 3 different hospitals, right? And these 3 data set basically can have different genotyping platforms, right? So the data set can have the differences inherent in a genotyping generation. So because of these different generations, it gives you the differences in terms of the genotyping quality, and it shows in a genetic association testing. So we collectively call this situation is batch effect, right? So batches is like, which the pipelines, they generate the data set, and the different the pipelines or workflows can have the differences in terms of quality information or technical artifacts, right? So batch effect is commonly discussed in a genetic study, but also in other biological research as well. And also other non-confounders. So one very best example is about the confounders in a association testing is the ancestry. So ancestry differences, I mean, we learned from the session 3 is, the population has their own structure in a genotyping information, and those populational differences can give you different results in terms of genetic association. So, think about the null hypothesis in a genetic association is that the biological differences doesn't make any changes in your experiments. So you are basically testing that hypothesis. But if there is any differences coming from other factors like ancestry, then you cannot compare the true differences or originate from the biological region, which is like disease status or trait differences, right? So in that case, you have to control those confounders for your association testing. And after that, once you find the gene or the variants from certain trait or disease, then you do the experiments after that. So after that means that the post hook, right? And then those analysis is normally containing functional evaluation. So you can put it into the cell, so in vitro, or you can put it into animals, which is in vivo, right? So you do sort of this functional evaluation approach. Then also you can check the reproducibility. So whether your result is, reproduce the previous findings or some differences. Okay, so talking about the sample size, this is one of the critical things in genetics. The reason is that statistics is basically describing the level of association in a certain trait or disease. And when we're talking about using statistics, we are basically calculating the probability of the effect size of the variance in certain trait. So when we conduct the experiments, we basically take some samples out of the entire population, right? So this is the population, let's say this is the all individuals of depression patients in the world. And if you do the research of the the depression, then you take out some of patients out of the population, right? So these small sets of people only describing certain aspects of the entire population. So when we're talking about the depression, let's say there are millions of people in the world affected by depression. Then if you study some depression in our country, then let's say you take out 300 people. Those 300 people might not be well representing the characteristics of depression in the original population. So when you conduct the genetic study, but also the biomedical research, you always have to think about the samples and make a sort of good estimation out of this sample size. And when we're talking about the sample size, we are also can think about the effect size, which is the magnitude of the effect of an allele on a phenotype. This effect size, we basically measured by odds ratio. We are covering this term in the next slide. So genetic associated studies is basically measures a effect size of genetic variants of your interest. So in this case, it's again, this is the describing the limited numbers of the entire population. So the sample size is critical part of estimating those statistical information, which is the effect size. So odds ratio is one of the way you can represent the effect size. So one example is that if you look at the case control study, the frequency of particular allele or haplotype is compared between the selected affected individual, which is the cases, and controls, which is the unaffected individuals. And you can measure an association between disease and genotype, then this is calculated by odds ratio. So you have the affected individual, which is we call patients, and you have the unaffected samples we call controls, and you're measuring the genotype information from both. And then you look at certain genotype, and we call it genetic marker. And this genetic marker could be a single variant or a single genotype or single locus. It depends on the experimental design, and we collectively call genetic marker. And you can count how many people has this genetic marker, which is genetic variant in a patient, and you put the numbers. And also likewise we do for controls, and then you count the number of patients without this genetic marker, then you put C, and likewise for the controls and D. Then you can calculate the odds ratio. So you use the C divided by D and A divided by B, and simply you put A times D divided by B times C. Then these numbers can be calculated how far this genetic marker associate with these patients. So I mean, in other words, associated with this disease, right? So your null hypothesis, which is H0, has odds ratio equal 1 because the all numbers should be equal to 1, or at least the patient and control should have the same, ratio between them, right? But then if you find the high odds ratio, which is over 1, then that means that you have some, interaction between this genetic marker and phenotype or disease, right? And you have higher odds ratio, that means the stronger impact on phenotype. So you can say your odds ratio is like 2, and then we can say, this is obviously some strong association with phenotype, right? This is one example of the exome study of autism spectrum disorder in a 2012 Nature paper. This is the very early days of exome paper of the autism research. So you read the paper of the Setterstrom 2020 in Cell. That is the latest one, and this is one of the first research. So if you look at the data set, so what you read in a Setterstrom paper in your assignment, you probably see higher enrichment of protein truncating mutation in autism cases. And is this true in early days? Yes, it is. And de novo nonsense mutation, in other words, this is part of the protein truncate mutation, right? And this red bar is the rate of the de novo nonsense mutation in cases, and blue bar is the ratio in controls, right? So odds ratio between the case and control in terms of de novo nonsense mutation is 5, right? And de novo missense mutation odds ratio is 2. And those differences is not observed in other categories, right? So this is the one of the revolution in a autism research in 2012, because not just study, other 2 additional study came up with the same conclusion, and they totally changed the perspective and paradigm in terms of the autism research in 2012. And then now people looking at the de novo nonsense mutation in autism patients, right? The things you can compare and you can find intriguing is the reproducibility, right? So reproducibility, you can ask whether the findings is the similarly observed in a recent study, or similarly observed in terms of the effect size, right? So de novo nonsense mutation, in other words, de novo protein truncate mutation, are the same evidence as these days, right? So the research between 2012 and 2020 is the same, right? But then the effect size, odds ratio here is the 5. If you look at the Setterstrom paper in 2020, the odds ratio is way down. So that is like a 1.8. So in terms of the effect size, the early study is quite overestimated compared to the recent side. This is the critical aspect, because always the early genetic studies, they have the overestimation in terms of their association testing. The reason is that they have much smaller sample size, and also they have very ascertainment in terms of the sample collection. So they tend to collect more severe phenotype and more distant individuals in their cases data set. So if you look at the genetic study in our history, always the first the evidence of the genetic association is the overestimated, right? So this is one implication for you. And another study from the type 2 diabetes exome sequencing, published in Nature 2019 quite lately, and that is giving you the association between the type 2 diabetes patients versus controls, right? So why I show you this example is that, like, you know, these mutations, like GLP-1R genes mutation or IGF-1R mutations, they are highly, you know, found in cases, right? Because the odds ratio is greater than 1. So this is the same situation as the autism research. But then some of the genes are enrichment, like an S5-2 has the odds ratio lower than 1. And this is same for this gene as well. So that case, we call protective alleles, right? So protective means that if you have the mutation in this gene, then you are unlikely to have type 2 diabetes, right? So type 2 diabetes is, you know, compared to autism, you can represent the phenotype in a bi-direction. So either you are, you know, prone to type 2 diabetes, or you are protective to or against the type 2 diabetes. So in that case, you can measure the bidirectional association. Then these things are useful in terms of, you know, you can predict the risk of having certain disease, not just type diabetes, like a cardiovascular traits or, you know, other, you know, some inflammatory diseases. So people want to find sort of the protective alleles, because some, you know, individuals obviously quite, you know, unlikely to have certain types of the disease, and people try to find why, right? Because this is quite useful information in terms of developing drugs, because, you know, like, you know, SLC5A2 is protective allele to type 2 diabetes phenotype. Then people focusing on, you know, this pathway and think about, you know, interventions or drug development, right? So all of this information is, again, people want to look at from the latest genomic information. Okay, so when we're studying genetic research, we can also coming up with key terminology, which is cohort. So cohort is one of the terminology you might see many times in the large scale genetic research. So cohort is referred to a sample of the population. It's selected and observed for some times to ascertain who does or does not develop disease, right? So you're not just selecting, you know, the patients or controls, right? Because, you know, people can have disease at a certain point or, you know, certain, you know, age, right? So people basically tracking those information over the time in a certain population. So in that case, you specifically call cohort, then you collect the information and tracking down whether those people has the disease in a certain time point or not. And this is obviously impacted by genotype or environmental factors, right? So cohort may be selected randomly or sometimes selected in a certain purpose, right? So we say targeted. So if you look at the some examples, I put it in here, like a Dennis blood spot. This is the data set collected all the blood information when baby born in Denmark in 1990, and they store that blood information and they tracking down their health information over 20 years. And if you find certain conditions or certain disease, then you back to the the blood information and do the genotyping and see whether there's any mutation associated with this patient, right? And likewise, you have other types of the information like an Avalon study here, or the Simon simplex collection. This is only targeted for the autism research, or you can select it randomly like a NOMAD study as we discussed previously. And also, other than cohort, we also can look at the family, right? So family is obviously, you use the kinship information and you can have the expected ratio of genetic factors within a family relationship. And this is easy way based on the IBD information, and you can easily track down how the risk genetic factors segregating in a family. And there's some notation describing the pedigree in genetics. So if you are super keen on the clinical genetics or diagnostic field, then you can read the manual. And some of the basic notations describing the family in genetics, like you probably found in a Setterstone paper, like a proband or propostus. I don't use that much, but proband is quite frequently used, or the siblings, right? So proband is the family member with the disease and a pedigree, and siblings is their brothers and sisters. And consanguineous is the couples who have one or more ancestors in common, right? So describing the cousin marriage, or if the proband is only affected in members in family, then we can say isolated cases. Or you are also called the sporadic case. This is more frequently used in these days because this is the describing the same situation, but we know the obvious cause, like mutation to the proband. So in these days, when you describing like a trio family, trio is like two parents and you have only one kid, and parents does not diagnosed by certain disease, but your child diagnosed by disease, then in that case you call this sporadic case. And this is the typical way you put the familial information, and you probably seen from the general biology about the disease inheritance. And this is again very easy description about the basic pedigree patterns, like an autosomal dominant, autosomal recessive, X-linked dominant or recessive, and this is by autosomal chromosomes or sex chromosome. And we know about the IBD situation, so we can easily think about the probability of segregation of certain genetic factors. Okay, this is the first part of the session on this session, and we have 2 more sessions to go. We are continue to discuss in next Wednesday. So up to this point, you are covering on a Monday session, and we are continue to discuss from the method session for association testing in a next time. See you later.